Hello everyone, welcome to today's live broadcast, Development of Paper-Based Zika Virus Test. I'm Robert Castellanos of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of your slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the C button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would like to introduce today's speaker, Dr. Keith Pardee. Dr. Pardee is an assistant professor in the Leslie Dan Faculty Pharmacy at the University of Toronto. He holds honors of Bachelor of Science degree in Biological Sciences from the University of Alberta, a Master of Science degree in the Natural Products Chemistry from the University of British Columbia, and a Doctor of Philosophy degree in Molecular Genetics from the University of Toronto. Following his completion of his doctoral studies, Dr. Pardee completed a postdoctoral fellowship under the supervision of Professor James J. Collins at the Wyss Institute at Harvard University. I'll now turn it over to Dr. Pardee for his presentation.
Hi, thanks, Rob. As Rob mentioned, I'm at the University of Toronto, and today I'm going to talk about how we use our cell-free synthetic biology applications to develop Zika diagnostics, as well as at the end, I'll touch on some new work we've done in portable drug manufacturing. Before I get started, I'd like to acknowledge all of the authors on the Zika virus paper, which is a very big project that we did in a short period of time. And so there was a lot of people involved in the success of this project. The project was led by James Collins, who's at the Wyss Institute at Harvard. So here's just a brief overview of the talk that I'll be giving. I'm going to start out with some background on synthetic biology, and then I'll get into how we developed the paper-based synthetic gene network platform, and then I'll talk about the development of that platform into a specific Zika virus diagnostic. So synthetic biologists are aimed to sort of rewire and reprogram cells with the goal of creating new sources of green chemistry for the production of commodities and therapeutics, as well as biofuels. And to do that, they are turning microorganisms into living factories. And they do this by placing engineered metabolic pathways into these organisms that produce the commodities, basically. And regulating the flux through these pathways are often synthetic gene networks. And a synthetic gene network is just simply a genetically encoded tool. There is a sensor component that can detect small molecules like metabolites, contaminants, environmental cues. There's a simple logic component and a measurable output. And that simple logic component can be configured into AND gates, OR gates, or just simple transduction, as is the case in the Zika virus diagnostics that I'll talk about, where basically if you have an input, you'll get an output. And so fortunate for synthetic biologists, nature is very good at sensing. And in fact, life depends upon it. Finding food, avoiding danger, maintaining homeostasis all require sensors on the molecular scale. And these synthetic biologists have taken these, embedded them into these networks. So now we have synthetic gene networks that can detect contaminants like heavy metals, pesticides, drugs, and nucleic acid sequences, which is the focus of the Zika talk, or the Zika diagnostic that we've developed. So you can imagine that synthetic gene networks have a lot of potential outside of simple bioproduction of commodities. And we envision these networks being useful in medicine, where a physician could use these networks to determine the source of an infection in a patient and to dig deeper to figure out what other clinically relevant pieces of information are there, such as antibiotic resistance. And the same paradigm can be applied to food safety and environmental monitoring. The challenge is that, by and large, most of these synthetic gene networks are housed inside microorganisms like bacteria, yeast, and algae. And in no way are those compatible with applications in the hospital, the food industry, or release into the environment. And so, recognizing the potential of these synthetic gene networks, what we did was develop an in vitro system, basically a material that's embedded with these networks, and the material can host these networks, and they're freeze-dried and stable, and they can remain sort of in a poised state, in freeze-dried states that can just, you add water, and they're stable and active for over a year. And so I'm going to talk now about how we developed that foundational platform. So to start, basically what we did was we used this in vitro transcription and translation system, which has been used in the lab as a tool to study transcription and translation for decades. And it works very well, and it's been really instrumental in understanding the molecular mechanisms that underpin these processes. But for us to deploy this out of the lab, there's some challenges. First of all, it's a liquid reaction, and so without pipettes and other specialized equipment, it's difficult to handle these reactions outside of the lab. And the other is that these reactions are very labile. They require being stored at -80 degrees Celsius, and so are difficult to bring out of the lab. And so, inspired by things like pH paper and chemistry-based diagnostics, such as that are on paper, such as those out of the George Whiteside's lab, I thought, well, perhaps we could merge the two ideas. And so, could we put these cell-free systems, synthetic gene networks, into paper and host them that way? And so, on your right, what you see is some paper discs that have been embedded with these enzymes of transcription and translation. And then the disc on the right also received a DNA plasmid for green fluorescent protein. And while the other disc did not. And then you incubate at 37 degrees, and within 20 to 30 minutes, you get the expression of this protein. So this was a great start. It worked. But of course, there were a lot of other components that need to come together. And one of those is the synthetic gene networks themselves. And I'm especially excited about RNA-based components for this role. The state of the field a few years ago was really focused on this type of riboregulator. And I'll walk you through sort of how they work. On the left side is a red box, which so the whole piece is a piece of DNA. The red region encodes the cis repression sequence, and the blue region encodes the ribosomal binding site. When transcribed into RNA, the cis repression sequence folds over and binds to the ribosomal binding site, blocking ribosomes from binding and therefore preventing translation, in this case of GFP, unless you add another component. And so here the DNA is transcribed, you get a piece of RNA, and that RNA has a higher, this is called a transactivator, and it has a higher affinity for that cis repression sequence than it does for the ribosomal binding site. And then as a result, the ribosomal binding site becomes exposed, ribosomes bind, and you can get translation. And so this is actually, I think, really cool. However, if you want to turn these into diagnostics, you can imagine there's some serious limitations in all. Basically, your transactivator, or the trigger that you're trying to detect, will always have to have some kind of complementarity to the ribosomal binding site. And so that's not very useful as a diagnostic. And so my collaborator, Alex Green, came up with the bright idea of moving that ribosomal binding site to the end of a hairpin and combining it with a molecular feature called a toehold. And so how this works is basically the region of that toehold switch, that hairpin that you see there, that region that's labeled AB is the sensor surface. And because the ribosomal binding site has been moved to the loop, there's very little constraint of what sequences you can put in there. And so the trigger RNA, A star binds to A, which is much like the tab on a zipper, and starts to unravel the whole hairpin, and B star binds to B. And the result is you get a linearized piece of RNA, double-stranded RNA, and the ribosomal binding site no longer has that steric strain on it, preventing the ribosome from binding, and you can get translation. And because there's few constraints on what can go into that AB region, you can rationally design these tools, and you can design them to complement synthetic RNAs as well as endogenous mRNAs, including those of pathogens. And so this is another early experiment that we did, where basically we have 7 different toehold switches on 7 different pairs of paper. All of the pieces of paper have got the enzymes of transcription and translation embedded into them. And they're freeze-dried, and basically we rehydrate them with either water alone or water plus the RNA. And you can see that in the presence of the correct RNA trigger, each of those toehold switches turns on to varying degrees. And you can see, because these pieces of paper are 2 millimeters in diameter, they'll fit into a 384 well plate. And you can put these and run these in a plate reader and get your quantitative data. And in some cases, very high on-off ratios and very digital responses. So great tools to work with. But of course, there were more features that were critical to make this platform field ready. And one was, as I've alluded to a little bit, distribution without refrigeration. And so for that, we turned to freeze drying. When we freeze dry these paper-based reactions, they remain active for over a year, and the cost per sensor is about 3 to 68 cents. We also wanted to be able to have colorimetric outputs. And so here, we want to be able to, in low resource environments, we want people to be able to read the output of their diagnostic in the absence of power or sophisticated equipment. And so here, there's a color-based output that allows that. And then finally, for more sophisticated applications, we have a low-cost electronic reader that could allow quantification and automation. So how did we create the colorimetric output? I tested probably 10 different combinations of reporter genes and color-based systems, and in the end settled back on LAC-Z or beta-galactosidase, which is a very common reporter enzyme in biology, likely because it works so well. And it worked very well. And basically pairing it with CPRG, which is the molecule at the bottom of your screen, the enzyme cleaves along that ether group and creates basically cleaving the molecule, turning the yellow substrate into a purple chlorophenol red molecule that has the absorbance of at 575 nanometers, which is important for the electronic reader that I'll talk about shortly. But you can see clearly there is a color change that's visible to the eye as well. If we sample those paper discs over time, you can see that the color change starts around 25 minutes and saturates around an hour. And just taking these discs, the series of discs alone, their images, and separate those red, green, and blue channels, you can see that the camera on your cell phone could in fact serve as a companion electronic for such a device. And these are the graphs you see here are the data that's been pulled out of the discs on the left side. And so finally, the electronic reader. This was built by Tom Ferrante, who's been a great collaborator, and he's sort of our go-to device electronics imaging guy. And he built this device using a laser-cut acrylic housing, which you can see a little low resolution, but you can still see the box that's been constructed, and then a green acrylic chip that has holes in it. And that chip, each of those holes contains a piece of paper that, when slid into the machine, places those pieces of paper between an LED that has a very narrow bandwidth at 570 nanometers, an electronic sensor at the bottom. And so as this purple color is produced, it blots out the transmission, and the sensor detects that change in relative transmission and creates these graphs. So at the bottom on the right, you can see some example data. This is synthetic switch G with various amounts of trigger, and you can see we get a dose response curve, and the line thickness sort of corresponds to the standard deviation. So you can see there's not a lot of variation, which is very nice. Our first reader cost under $100, and the readers we're using now, which have capacity for much more, many more samples, are under $250, and have lithium-ion batteries so that they can be deployed into remote regions where there's no electricity, and they have onboard data storage. That makes it very easy to analyze the data afterwards. So to summarize this part of the talk, which is the introduction of the base technology, by taking these synthetic gene networks out of the cell and embedding them into paper along with these cell-free systems, we've created programmable and responsive materials, and hopefully a new venue for synthetic biology. One of the big bonuses of this approach is that you have very short design to test cycles, allowing us to prototype rapidly. And that capability was really important in our work with the Zika virus. The applications that we're focusing on are sensing or diagnostics, and on-site, on-demand manufacturing of therapeutics, which I'll talk about at the end of my talk. So moving on to the Zika Project. At the end of 2014, we published this first work on paper-based synthetic gene networks. We built some sensors against antibiotic-resistant genes and Ebola virus, which was really exciting because this hadn't been done before. But the practical, sort of implications were that we weren't sensitive enough yet to be clinically relevant. And so during 2015, we worked hard to improve the user sort of interface, as well as make these sensors more sensitive by developing amplification schemes in front of the toehold switch. And so with the goal of having a scheme sort of like this, where you have a freeze-dried system in paper that contains your synthetic gene network and is rehydrated by the diagnostic sample, essentially producing programmable diagnostics that could be fairly rapidly applied to any disease, just because it's all sequence-based, and that the toehold switches are rationally designed. And so in early 2016, when the outbreak of the Zika virus in Brazil was first reported, Jim Collins phoned Alex at his new faculty position in Arizona and me in Toronto, and suggested that this was an opportunity for us to contribute to this outbreak by using the toehold switch and the paper-based diagnostics to bring molecular diagnostics into the field, which was key for the Zika virus. Basically, the Zika virus, for various reasons, requires molecular diagnostics to provide really clear diagnosis. And so we immediately said yes and put projects on the side, and so did a number of other people, a lot of other people. And the team assembled rapidly, and we got to work. And so the first thing was to find a genome for the Zika virus. And at that time, Alex, there was only one available for the American strain of the Zika virus, and Alex found it on the NCBI database. And using his algorithm, walked through the genome, finding regions of the genome that had low secondary RNA structure and also were single-stranded, basically regions where the toehold switch would have access to the genome to bind. Then he cross-referenced that against closely related viruses like the dengue and chikungunya to make sure that we would not get cross-reactivity with those viruses, and then against the human genome to make sure that there was no issue there. And then when the algorithm had completed that process, there were 24 regions of the genome that had been targeted, and the complement of each of those regions, labeled here A through L, basically was placed into the toehold switch in that place where that bold line is. So you can see that this is a very simple way to build sensors. And with a development cost where our DNA costs about $20 and our manufacturing costs about under $1, you can see that while, of course, antibodies are very sensitive and certainly have a role in many applications, this new approach is very fast and has low cost of development. And so it has some really compelling features for rapidly responding to outbreaks. And so just to recap the development phase, basically the algorithm designs the sensors and then DNA synthesis produces them. This takes about 4 days. Once we receive the DNA in the lab, it took about 7 hours to assemble 48 sensors. So this was 2 different types of toehold switches targeting 24 different regions in the genome, and validate them in the cell-free system. This took 7 hours, and then we give ourselves a day for manufacturing and distribution to start, just as a sort of guide. So this is what the first data looked like from that 7-hour experiment, where we assembled the sensors and then tested them. And you can see, I'd mentioned there's 2 different types of toehold switches. We call them the A series and the B series. The B series provided a slightly higher output. And if you look at the inset of that graph, also a lower background. We took the top A series and the top B series sensors and tested them to a titration of RNA triggers. And as you can see, we really couldn't detect much below 30 nanomolar, which was not unexpected. As I mentioned, during the year 2015, we spent the time trying to build up tools for how we could reach clinically relevant concentrations of detection, which for the Zika virus is 6 to 7 orders of magnitude lower, so in the low femtomolar range. And so work done by Melina Fan and Dana Braff had been incorporating an isothermal amplification scheme called NASBA into the workflow. This is a primer-directed process where primers target regions, those 24 regions in the Zika genome, to create basically a double-stranded piece of nucleic acid where you have a new strand of DNA matching the RNA sequence. That RNA is then degraded by RNA-Lych, and another primer is used to add T7 promoter to this double-stranded piece. And now you have a piece of DNA that basically serves as a photocopier that can produce lots of copies of the RNA rapidly. And this cycle continues, and you get amplification, and it's from this that we use to rehydrate the toehold reaction. And so when we now place this amplification scheme in front of the toehold switch, we can very easily detect down into the low femtomolar range. You can see here we've used RNA in water or RNA in a fraction of human serum to sort of replicate the diagnostic, the ultimate application in diagnostic as a diagnostic. So we've now got the development of the sensor itself, an amplification scheme, and now we needed to develop a fieldable method of extracting the RNA in sample prep. We tried a number of different methods, and in the end settled on a very simple one, which was heating the virus sample up to 95 degrees for 1 or 2 minutes to lyse the virus and release the RNA. And that worked really well. In 1 or 2 minutes, we were able to, basically the toehold switch was able to, we'd amplify, we were able to amplify the virus rapidly, and then from the virus, or the RNA from the virus, and then the toehold switch could see the RNA. So what I'm describing here, we've gone from synthetic RNAs that coded for regions of the genome, and we're trying to build complexity and slowly build towards the type of sample that you'd see in a clinical lab. And so our next step, which is the graph on the top left, was taking viruses that were grown in Lee Gerke's lab at MIT, so 3 isotypes of the dengue, as well as the Zika virus, and extract the RNA from those using the heat-based system and test hit on the toehold switch to determine how specific our Zika diagnostic was compared, and was it basically, could it just specifically detect the Zika? And so using RNA that came out of those viruses through heat method at 30 femtomolar, you can see that we had great specificity and strong signal for the Zika test. And taking it a step further, we had samples that were generously provided by Dave O'Connor's group at UW-Madison. They're working on the effects of the Zika virus on physiology, and are doing so by working in monkeys. And so they sent us viremic plasma from macaques, and that had the viral concentration of 2.8 femtomolar. And so following heat treatment, isothermal amplification, we were able to detect the virus with the portable reader. This is all done with the portable reader, very, in under 30 minutes. And so the amplification scheme here was 2 hours or 2.5 hours, and we get detection, very clear distinction between the positive and the control sample. And so in the end, we've been able to use this paper-based platform in the toehold switches to build diagnostics that can detect in clinically relevant concentrations and detect all global strains of the virus. We can detect with these sensors, they're capable of detecting the American strain, the Asian strain, and the African strain of the virus. And importantly, also discriminate between the strains of the virus. As you may know, the American strain of the virus is associated with rare but very severe neurological symptoms. And so it's important to be able to discriminate between these strains. We're now at a point where this project took about 6 weeks to produce. We now have, with this pipeline sort of established, we can now do this in about a week now. And cell-free systems certainly play a key role in accelerating that prototyping. I'm now going to talk about our ability to distinguish between the strains of the virus. And this was an idea that was generated by Guillaume Lambert, who was a postdoc also in Jim's lab and is now a faculty member at Cornell. And Guillaume developed a system based on a module CRISPR-Cas9 system. And what he did was he looked at the 24 regions of the Zika virus genome that were targeted by the toehold switch. And he looked for what are called PAM sites. PAM sites help align the Cas9 protein on the genome, and they're basically a nucleotide followed by 2 Gs. And what he was looking for was a PAM site that was different between the 2 strains. And so if you look at the bottom right, I'll explain sort of how it works. Using isothermal amplification, primers add on a synthetic trigger, which we call H, and basically get a double-stranded piece of DNA, just as we did in the original NASBA reaction, except this time there's that double-stranded piece at the end in yellow that is the trigger. And so in the presence of the full PAM site, which is in the American strain here, Cas9 recognizes it, binds and cuts, and you don't get that trigger region transcribed into RNA. So you don't get the toehold switch H being activated. But in the case of the African strain, where that PAM site is not present, Cas9 does not bind, you get the trigger H encoded into the trigger, or the RNA, and you do get sensor H, which is a synthetic orthogonal trigger activated. And so through this method, we can get single base pair resolution differences between the strains. So just to recap, this system allows us to take an unknown sample, virus sample. We can detect all the global strains of the Zika virus while discriminating from closely related viruses like dengue. And then we can also distinguish between the American and the African strain. And in this case, the PAM sequences result in the American strain having a yellow color, and the African strain having a purple color. And so just to recap, we have we developed using the system a portable low-cost diagnostic platform for the Zika virus that can discriminate between the viral strains using a CRISPR-Cas approach. We can detect down into the low femtomolar range of the virus from monkey plasma, which is in a clinically relevant concentration. And we developed a programmable sensor workflow that could be used really for any rapid response to an epidemic. Our next steps are to incorporate low-cost microfluidics for sample prep, and we're working with partners towards field trials in Brazil, Ecuador, and Colombia. So far what I've talked about has focused on diagnostics, but we could also use the same cell-free freeze-dried system to produce therapeutics in a portable way. And the therapeutics that we're going to be talking about are largely protein-based therapeutics that require cold chain for distribution. And this work was published in September. And before I talk about it, of course, I'd like to acknowledge all the people that were involved in this project, which was also done very quickly with a large effort from all of these people out of Jim's lab again at the Wyss Institute at Harvard. So just sort of to give you a real overview of what we're doing here is instead of a synthetic gene network at the top of this flowchart, it's just an expression construct. And instead of a paper-based reaction, it's a freeze-dried pellet in a tube. And basically, with the right construct, you can produce lots of different types of therapeutics. And I liken this to basically bringing the ease of 3D printing to the molecular level. So just as 3D printing can result in allow people to produce lots of different objects that normally would require high level of specialized skill, this approach allows people to make molecular tools and therapeutics literally in their pocket with just being able to add water to rehydrate reactions. And so we see this as a potential very useful thing to produce therapeutics, especially protein-based therapeutics that usually require refrigeration for distribution and storage to regions that have low resources and are remote. They could also provide low-cost reagents for research labs and clinical labs around the world. And in vaccine programs, sometimes, such as the flu virus, the strain composition is incorrect. And this could allow for this distributed capability of manufacturing could allow for repositioning vaccines efforts more rapidly. So just to give you sort of an overview of what we did, we took the cell-free systems and put them into pellets, freeze-dried them. Same thing with the DNA. These can be shipped at room temperature and distributed, and then on-site, on-demand manufacturing by simply rehydrating them. And you can produce these research tools and therapeutics. One of the most compelling areas, I think, for this platform is in the production of vaccines, which have a huge public health benefit. But of course, because they are protein-based therapeutics, in many cases, not all, require refrigeration. And this creates challenges, especially in parts of the world where refrigeration is not as available. And so we produced 4 different vaccines using this platform, one for anthrax, one for one for botulinum, and 2 for diphtheria. And with injectables, one of the concerns would be that since we're using a bacterial system for expression, is what are the risks of injection and what are the endotoxin levels? And basically, the threshold for toxoid-based vaccines is 200,000 units of endotoxin per mil. And when we purified our DT5, which is the diphtheria vaccine, we had orders of magnitude below that, 596 units per mil. So that was fine, as well as when we injected them into the mice remained healthy and happy. Did not seem to have any adverse reaction to these products. We had habitat control mice, which are on the left. In the middle, we had mice that were injected with purified product from cell-free reactions that did not contain DNA. And on the right, the purified product, the DT, that cell-free reactions that contain the DNA coding for the DT of the diphtheria 5 vaccine. And you can see around week 3, induction of anti-diphtheria IgG response, which corresponds to the production of antibodies or vaccination in the mouse. So we're pretty excited about this, being able to produce a vaccine from in a portable, sort of compatible fashion from a freeze-dried sample. Another application that we did that I'm really excited about is our ability to make antibody fusions or conjugated antibodies using the cell-free system. And so how we did that was on the left side of your screen, we had a library of antibody mimics, such as DARPins, and antibody single-chain antibodies, such as nanobodies. And on the right side of your slide, we had a library of functional groups. So these are things that you might want to append or attach to your antibodies, such as fluorescent proteins, alkaline phosphatase, other enzymes, or toxins. The antibody mimics, or nanobodies, had what's called a SPI tag, and the functional groups, or fluorescent proteins and enzymes, had a SPI catcher. And when these see each other, they form a covalent bond that essentially creates a single molecule. And so we can express each of these domains separately, and then post-translation, they can fuse to form a larger complex molecule. And so this scheme worked very well. We produced 12 different nanobodies and 3 different DARPins, and the antigens to which they're specific are listed on the right side beside the anti-flag Western, which is labeled B. And below that, with the Kamasi gel that's labeled C, are the bands that we produced that are the output domains. So this is the fluorescent protein we used, YFP, enzymes, which include alpha amylase, 2 different types of alkaline phosphatase, and then cellular toxins. So one from Pseudomonas aeruginosa and another from diphtheria that could be used as therapeutics for targeted cell killing. So when the nanobody and in this case, this is an anti-flag Western, with a nanobody against CEA5, which is a colon cancer epitope. And you can see that, that's the band at the bottom of the Western, between the 20 and 15 kilodalton marker. And then you see, as you go up, you see a second set of bands, which is a dimer, which happens sometimes with nanobodies. And then these variable bands that have arrows are the fusion products. And so here you have, in lane 1, you've got CEA5 fused to YFP. And as you go across, you can see the different products that we produced. So the next step was functional validation of these. And so here, this is a dot blot, which you can think of sort of as an analog to a Western blot, with 4 different nanobodies, one against an epitope from HIV, GFP, rotavirus, and C. difficile. And then with antigens spotted in rows, each of those spots were probed with these nanobodies fused to alkaline phosphatase. And you can see that we get light generation from the alkaline phosphatase in the spots on the diagonal, as you would expect. So it worked very well. We also wanted to try using this approach to produce products for fluorescent imaging. And so what you're seeing here on the left side are 2 different cell lines, one that's HER2 negative and one that is HER2 positive. HER2 is a breast cancer surface marker. And using our anti-HER2 DARPin, we were able to probe those cells, and then using a commercial anti-flag antibody with a fluorescent label, you can see we could label these cells fluorescently by combining these commercial and in-house produced reagents. But we could also do the same thing with the anti-HER2 DARPin that was fused to YFP, and you can see the staining here. As you go down, you see the DAPI and then the overlay of those images. And then these being a marker for breast cancer, we thought, well, could we selectively kill these cells using an fusion that is linked to a toxic protein? So we used an exotoxin from Pseudomonas and and targeted it using the anti-HER2 DARPin. And you can see on the far right of the column graph that when we diluted that cell-free reaction by 1 in 500 or 1 in 1,000, we got selective killing of those cells. And as a final example, C. difficile infections, which affect the intestine often, one of their sort of main clinical problems that they cause is they secrete a toxin that causes a lot of the sort of clinical symptoms that are associated with the infection. And using an anti-C. difficile nanobody that binds to that toxin, we were able to, others have shown, but we were able to produce that same nanobody in the cell-free system that binds the toxin and neutralizes it. And so in cell-based assays, you can see that on the y-axis here, you've got cell rounding. And so along the top are where all the cells, cell rounding indicates cell death. And as we increase the amount of the cell-free reaction that express this nanobody, you can see how we reduce the cell rounding, or the basically cell death, down to 0. And images along the bottom reflect this. So on the left is are the cells in culture exposed to just PBS, basically, with no toxin. In the middle, cells exposed to toxin, and you can see the cell rounding, which is indicative of cell death. And then on the right is the nanobody against the toxin plus the toxin, and you can see we don't get the cell rounding. And basically, you get rescue of the cells from the toxin. So to summarize this part, basically we use the cell-free system here as a platform for on-site, on-demand molecular manufacturing of therapeutics and lab reagents, and has the potential to provide distributed production of therapeutics that can overcome challenges that protein-based reagents and therapeutics need, which is refrigeration. And although I didn't talk about it, we also demonstrated, in addition to vaccines and conjugated antibodies, we demonstrated the cell-free platform with antimicrobial peptides, as well as small molecules. So to sort of summarize the whole talk, by basically taking synthetic biology out of the cell and putting it into materials like paper, in conjunction with the cell-free reactions, we've been able to create cell-free diagnostics based on synthetic gene networks and a portable molecular manufacturing platform. So in closing, there are a lot of people to thank. I'm grateful for all of their contributions, without which these projects would not have happened. I'm especially thankful to Jim Collins, who was my postdoctoral supervisor through these projects, as well as led both of these projects. And so with that, I would be happy to answer any questions, and I'll pass it back to Rob, or answer questions. Pass it back to Rob.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And we're going to try to answer as many questions as we can as time allows. So Doctor, our first question is, can you explain the freeze-drying process that you use for the paper-based diagnostics and manufacturing protocols, and how do they differ?
Sure. So basically we use sorry, I think we had a technical. There we go. So the freeze-drying process is very standard. It's the same process that is used by any other freeze-drying process for proteins, so there's nothing special about it. What differs between the two is basically, in one case for the diagnostics, we're freeze-drying onto paper, and the other case we're freeze-drying in a tube to create a freeze-dried pellet. And freeze-drying is basically done by flash freezing the cell-free reaction with its associated DNA constructs in liquid nitrogen, and then placing it on the lyophilizer and in a matter of hours, your sample's freeze-dried.
Thank you. And our next question, is ongoing mutation of the Zika virus an issue for the Zika diagnostic?
Great question. As you know, viruses and pathogens mutate over time. This is part of their strategy to avoid the immune response of their host. And so the virus does mutate over time. Fortunately, the toehold switch is very tolerant of mutation and can tolerate as many in that sort of 30 base pair window that the toehold switch probes, can tolerate as many as 4 different mutations. And so with this in mind, the toehold switches that we built theoretically have the capacity to detect all of the Zika virus strains that have been identified since the virus was originally identified in 1947 in Africa, in Uganda. And so, and we demonstrated that our switches could detect the Asian, African, and American strains of the virus. And so while mutation is ongoing and expected to continue, the toehold switches are expected to also be very tolerant of this and should not be an issue.
Great, thank you. And it looks like we have time for one last question. What were some of the challenges you faced in developing the manufacturing platform?
Okay, yeah. So the challenges between the two platforms were quite different. For the diagnostics, we were running toehold switches that essentially were, very minor changes in the sequence, but essentially the same. And so once we'd optimized the system for one, in many ways we'd optimized the system for all of them. In the case of the therapeutics, every protein is very different, and each protein or product that we produced, enzyme, required basically optimization to get maximum expression and maximum activity. And so it does add time to the production, but then once you're done, which takes a few days, or in some cases more, but in theory it can be quite quick. Your system's very stable, and you just had to define those features early on. Thank you.
And do you have any final closing remarks for today's event before we let you go?
Well, thank you so much for the opportunity to share my work today. And for anyone who's interested, please contact me by email. Our lab is growing, and we're very interested in having new students join us or postdocs in the lab as we take both these platforms, these cell-free technologies, towards sort of expand them out in the manufacturing and diagnostic space, as well as develop new technologies in this space.
Well, thank you again, Dr. Pardee, for your time, for your wonderful presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through February 2017. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's event. Until next time, goodbye.